Hello and welcome everyone. In this podcast we're going to talk about the extensions to Mendel's rules of inheritance. Now remember, Mendel did his research in the 1860s, and then he left his research behind and took on more administrative roles, and his work was, for the most part, unknown until 1900. In 1900, when his works were rediscovered, scientists from around the world started taking interest in this, and they started testing their organisms to see if their organisms followed these same rules. And it turned out most of them did. And yet there were some extensions, or some exceptions we might say. The word extension is probably the best word to use because his rules were still correct. He wasn't wrong. Mendel was still a rock star. We just had to put some extensions to explain some of the observations. Chromosomes were still assorting the way they were supposed to, and they were still assorting into gametes the way they were supposed to. It's just that sometimes when the genes interacted in certain ways, we got phenotypes that we didn't expect. So it's best to call these extensions to Mendel's rules. And what I want to do here is make a list of everything we're going to talk about. We won't cover this all in one podcast. We'll have to split this up. So the first thing we're going to do is look at dominance. There's going to be complete dominance. There will be incomplete dominance. We'll talk about codominance. And something that we call overdominance. We're going to talk about lethal alleles, talk about two terms, pleiotropy and phenocopy. We'll talk about sex-influenced and sex-limited traits. We're going to want to talk about how the environmental factors, so how environmental factors affect the trait. We're going to talk about this general concept called gene interaction. And in this category, we're mainly going to talk about this unique thing called epistasis. And then we want to talk about the difference between a discontinuous trait versus a continuous trait. We're going to talk about this first. I just forgot to mention it up here. We're going to talk about what a genetic polymorphism is. And these terms called, we're going to have to abbreviate because I'm out of room here, expressivity and penetrance. Now I'm going to break this up into probably 3 different podcasts, and then I'm going to close with one summary for all of them, because really these are all linked in this one large topic of extensions to Mendel's rules. So let's go ahead and get started with talking about genetic polymorphisms, expressivity and penetrance, and then talking about dominance. And then we'll move through the list. Alright, so the first thing we want to talk about are genetic polymorphisms. Up to this point, we've always talked about there being a wild type and versus a mutant. And these wild type strains have some sort of advantage over the mutant strain. For instance, we can talk about flowers. And I'll give my best attempt at a flower, but you will not be shocked to learn that my flower drawing skills are limited. And let's say that this red flower is an advantage trait. Maybe it attracts pollinators better, maybe it avoids herbivores better. For whatever reason, strains that are that are red, produce red flowers, have an advantage over flowers that produce white flowers. And I'm just outlining it in black here because I can't really draw a white flower on a white background. Let's say there's also a purple flower that also does quite well. In fact, there's no advantage to being red or purple. There's a disadvantage being white, but there's no advantage here. And there might even be a third one. Maybe there's a blue one. And again, no advantage between being red, purple, or blue. These are all coded by the same gene. These are all coded by the same gene. They have different mutations in them. This one gives red flowers, this one gives purple flowers, this one gives blue flowers. This one gives, say, white flowers. They have different alleles. No advantage to any of them. That's what a genetic polymorphism is. Poly meaning multiple, morphisms meaning different forms. So these three different forms are equally advantageous. There has been no selection over these three. Maybe in time there will be. So they're going to be found in equal proportions within a population. Next I want to talk about these terms called penetrance and expressivity. Now I'm talking about penetrance and expressivity. Let's take a step back and think about what Mendel saw when he had these two genotypes. Big Y, little y, big Y, big Y. He always saw yellow seeds. And while this is always true, this genotype is always going to give you yellow seeds, what we've learned since Mendel is that sometimes a given genotype may not always give you the same phenotype. We may get one phenotype, or maybe two phenotypes, or three phenotypes, some kind of difference within these phenotypes. There's still a general theme between these phenotypes. It's not that going from the difference between yellow seeds to a tall plant. They're still would all be in the range of seed color, but sometimes there's some differences in the way these phenotypes appear. One of the best examples to demonstrate both penetrance and expressivity is a condition called polydactyly. This is a condition when a person has more than 10 fingers or more than 10 toes. Polydactyly. So I'm going to erase this to make a little room for us, and let's restate this up here. The average person will have 10 fingers and 10 toes. Someone with polydactyly, as I just said, will have more than 10 fingers or toes. So let's talk about this term penetrance. Penetrance is defined as the percent of individuals in a population with a particular genotype that express the expected phenotype. And so let's give an example. So let's say that the average person who has 10 fingers and 10 toes, they have the genotype of big D, and then somebody with polydactyly has the genotype of little d, little d. We expect the people with big D we would expect 100% of them to have 10 fingers and 10 toes. And then somebody with little d, we would expect to have 100% of those individuals with more than 10 fingers or toes. But penetrance tells us that that's not always true. We may have less than 100% with more than 10 fingers or toes. For instance, if we were looking at 100 people who are all little d, but only 81 had polydactyly, then we would say that this trait of polydactyly in this population of 100 people is 81% penetrant. I'm going to say that again. In a population of 100 people who are little d, so they all are little d, only 81 of them have polydactyly. So we say that this trait in this population is 81% penetrant. The other 19% still are little d, but they have 10 fingers and 10 toes. So it is 81% penetrant. And talk about expressivity. Expressivity is the degree to which a trait is expressed. So if you recall, I've been fairly general about what the phenotype of polydactyly has been. It says greater than 10 fingers or toes. I haven't said how many fingers there are, how many toes extra there are. I've been pretty general about that. And that's because of those, say on their last example, of those 81 people, or it was 81% as well, with polydactyly, so those who are expressing the traits, they don't all have the same number of fingers or toes. Let's say that 20 of them have 6 fingers on one hand and 5 fingers on the other hand. Let's say, and other toes are fine. And let's say 30 of them have 8 toes on one foot, and that's the only thing different. And then let's say 31, the rest of them, have 7 fingers per hand and 8 toes per foot. These could be anything. I'm just making these numbers up. The difference is that all of these people are little d, yet they don't all have the exact same phenotype. There's some variability here. Alright, I'm going to erase this and say one more thing about these two terms. And the way we often refer to these terms in a population, and we'll keep talking about polydactyly here. In the example I gave you where 81% had the trait, but they weren't all the same as far as the way they expressed it, we would say that this trait had or has incomplete penetrance. In this particular case, we could say it's 81% penetrance, but sometimes you don't know the exact percent. You just know that within any given population that it's not going to be 100%. So we would say it's 81% penetrant if you know the number. If you don't know the number, you say it's incomplete penetrance. And in this particular population we've described, we would say it has variable expressivity. So why do we see this? Why do we see incomplete penetrance and variable expressivity in this trait of polydactyly, as well as in many, many traits? It's for two reasons. One is because of gene interactions. And what I'm saying here is that these traits aren't controlled by one gene. They're controlled by multiple genes. And these genes, when they make proteins, they interact in different ways. So they may have one or multiple genes in this pathway mutated. Or they may have them mutated in different parts of the pathway that gives you these differences in penetrance and expressivity. Another factor is the environment. Environment plays a huge role in many of these traits, including polydactyly, where depending upon the environment, it can change the penetrance or the expressivity. And let's talk about dominance. Up to this point, we've talked about traits that are, we haven't used the term, but this is what we meant, completely dominant. For instance, if we look at Mendel's plants, big Y, little y, or big Y always gave you yellow seeds. The Y in this case is completely dominant, and little y always gave us green seeds. We now know that not everything is completely dominant, and so within this area of dominance we're going to talk about a few other examples. So complete dominance is one, and I think we've already talked about that sufficiently. It's when one allele has total dominance over another allele. We're also going to talk about incomplete dominance, something called codominance, and something called overdominance. And sometimes this is called heterozygous advantage. Alright, I'm going to erase this and we're going to start talking about incomplete dominance. So for incomplete dominance, it's a trait where when you look at the heterozygote, so we'll go big A, little a, has a phenotype that is intermediate between the homozygous dominant, in this case big A, and the homozygous recessive, little a. The classic example that we always talk about when we talk about incomplete dominance is the phenotype associated with the color of flowers. And not all flowers, just this particular flower. We see it in, one example is snapdragon flowers. So you take a red flower and you cross it with a red flower, you get red flowers. If you take white flowers and you cross it with a plant with also white flowers, you get white flowers. When you take a red flower and you cross it with a white flower, they would have predicted originally that you got red flowers. But when they did this cross, that's not what they got. They got pink flowers. And this is the example. This intermediate, this heterozygote, is somewhere in between. Its phenotype is between red and white. So I'm going to redraw this in more of a cross format. So if we say the red allele is R1 and the R2 allele is white. So R1 gives us red, and R2 gives us white. So if we do a Punnett square here, we put R1, the red flower up here, and then R2 and R2, we end up with R1, R2. All of these are going to be R1, R2s. They're all going to be pink. Now, then if you take the F1 progeny here in the middle, F1 crosses, and you cross them to themselves, what you get is 25% of them will be red, R1, R1. 25% will be white, R2, R2. And then 50% of them will be pink, R1, R2, R1, R2. So as we can see here, as we're looking at this, Mendel's rules still apply. We still follow the genes as they're being segregated into gametes, and as they're forming progeny here, you follow the same rules once again. It's just that the interpretation of the phenotype is now different. We don't have a case where R1 is completely dominant to R2. Instead, R1 is incompletely dominant with R2. Now, in the case of an exam, it's unlikely I'm going to use this example. I'll use another example where you have to look at it and say that the heterozygote is intermediate, is somewhere in between the two extremes, in between the fully dominant and the fully recessive. Okay, let's talk about codominance next. Codominance is when you have two alleles, and we're going to call them A1 and A2 now. And these alleles, they are both expressed, so both alleles expressed. And in their expression, they're both, I should say, equally expressed, fully expressed, meaning you fully detect A and you fully detect A2. If you think about our example on the previous whiteboard, where we had R1 gave us red flowers, and R2 and R2 gave us white flowers, and R1, the heterozygote, and R2 gave us pink flowers. Well, if this were a case of codominance, the heterozygote R1, R2 would fully express red and fully express white. So you wouldn't get a blend, you would get a flower that maybe, for lack of a better way of drawing it, might look like this, where some of the petals were red, some of the petals were white. Or you might see something like this, where you had parts of it red throughout and parts of it white. Both phenotypes of each of these alleles are expressed. We see red and we see white. We don't see a blend. That's one of the best ways to tell if you're looking at codominance versus incomplete dominance, is to look at the heterozygote. If the heterozygote is a blend, red, white, then pink, then you would say it's incomplete dominance. If it's codominance, like we're talking about here, you would fully detect both phenotypes, both red and white. You would recognize white and you would recognize red. That would be codominance. They're sharing the dominance. Okay, let's erase this and talk about a specific example. And let's talk about blood types. And let's talk about the ABO system in humans. And for now we're only going to talk about A and B. We'll talk about O later when we talk about epistasis. But we're only going to talk about A and B for codominance. Somebody who is blood type A, we'll talk about that person, then we'll talk about somebody who is blood type B, and then we'll talk about somebody who is blood type AB. Oh, and we might as well put down this fourth one just so we don't forget. There's blood type O. And to keep you coming back for more, I'm going to tell you about another blood type that is something different than AB, or O, and we're going to call it blood type H. But you have to come back to learn more about that later on. Okay, but let's focus on A and B so we can explain codominance. Somebody with blood type A, their genotype is this. It's the same gene, but it's the allele A. Just like somebody who's blood type B still has the same gene, but it's a different allele. It's the B form of it. Somebody who is heterozygous has the gene IA and IB, and their phenotype is blood type AB. And this is the example of codominance. In the heterozygote, we detect folate A and we detect folate B. We don't see a blend. We don't see that A is dominant over B or that B is dominant over A. They share in their dominance. It's codominant. The last thing we want to talk about with dominance is something called overdominance. Sometimes we call this a heterozygous advantage. So we're going to say, for the definition of overdominance, is that the heterozygote has a reproductive advantage over the homozygote. Either the homozygous dominant or the homozygous recessive. The best example to talk about overdominance is to talk about the disease sickle cell anemia. Sickle cell anemia involves a mutation on the hemoglobin gene. So if a person has this genotype, HbA, they have average shaped red blood cells. They're oval. If somebody is HbS for sickle cell, HbS, same gene that just mutated, they have sickle shaped red blood cells. Somebody who is HbA, HbS, so the heterozygote, they have, for the most part, average looking red blood cells. There might be some sickling with them, but phenotypically they're pretty indistinguishable from HbA, HbA. So why does this matter? Well, let's erase this. So let's draw a blood vessel. Turns out that the average red blood cell is extremely flexible. As it's moving down a blood vessel, it'll enter a capillary. And in order to get through this capillary, the red blood cell has to take advantage of this flexibility so it can get through this tiny capillary. In the case of sickle cell anemia, these sickle-shaped ones are very rigid in their shape. And what happens is they can't get through this as efficiently. And they kind of get clogged up here because they don't have this flexibility. So this would be somebody who is HbA, or somebody who is HbA, HbS, the heterozygote. So there's no disadvantage to being a heterozygote in this case. Down here is somebody who is homozygous recessive for the sickle cell anemia trait. Because these red blood cells cannot move as efficiently to different organs, this disease of sickle cell anemia is characterized by an inability to efficiently move oxygen to tissues that need it. People who have sickle cell anemia, they'll have a series of health problems. They'll be more susceptible to infection. They'll experience pain, particularly in their joints. They may have chest pains, respiratory problems, and often will have a higher incidence of stroke. And because of this, people with sickle cell anemia have a lower life expectancy. So what is the advantage to being a heterozygote? Well, it turns out that the disease called malaria is caused by this plasmodium that enters into the cells, into the circulatory system, and infects red blood cells. And so it'll attack these red blood cells and kill them as the plasmodium completes its life cycle. Well, it turns out, in fact, let's erase the heterozygote here. It turns out that plasmodium is very efficient at attacking and killing the homozygous HbA. However, it can't infect the HB sickle cell. However, there's no advantage to this because that's a decreased life expectancy. There's no advantage to this if you have malaria. But it turns out if somebody who is HbA, HbS, is infected with this plasmodium, it can't gain access to the red blood cell. So there's this advantage. They now have a selective advantage. They will have higher reproductive success than the individuals who are HbA, or those who are HbS. Because the people who have sickle cell anemia, lower life expectancy because of the sickle cell anemia, so we'll be able to reproduce less often. In areas where malaria is high, in certain parts of Africa and South America and Central America, these individuals will be prone to the disease malaria. However, this individual here, the heterozygote, has a distinct advantage. The red blood cells are just as easy to move down these capillaries, so they're not going to have sickle cell anemia, but they have the added bonus of being resistant to malaria. And so this is why we see increased incidences of sickle cell anemia in parts of the world where malaria is high, because this allele, the sickle allele here, has been selected in those areas because it's advantageous if you are a heterozygote. So that is heterozygous advantage. So be able to recognize this if I were to ask you a problem, not about sickle cell anemia, but about another disease or condition where the individual had some sort of advantage as a heterozygote. Okay, I think this is a good place to end this podcast. We talked about genetic polymorphisms, expressivity, and penetrance, and we talked about the different cases of dominance. In the next podcast, we'll talk about lethal alleles.